we go, the faster we'll be able to survive the day. So I am Jasmine Aguado Sierra. I work here at BSc. I work on multi-scale computational modeling of the heart. So I'll be talking a lot more about the parts of the electrophysiology problem that we solve to approximate this, the simulations of the heart. So one thing that we do, or what I try to do here, is this new field of computational cardiology. There have been so many advancements in learning about biology, about pharmacology, about anatomy, about genetics, about so many things. And now the problem, we know many things about dividing the heart, for example, or the cardiovascular system in tiny little pieces. So what computers or what HPC gives us in this regard is that if we build all of those into mathematical models, and then we put all of those mathematical models into a software and into a computer, we might be able to understand all of these small pieces, how they interact together, and how they can help you predict or understand physiology, disease, or different things, or to apply all of these models on a more patient-specific manner. So that is what we're trying to do. We're trying to do this computational cardiology, where you look at all of these little pieces and put them together and trying to solve them in HPC. So it's a complex, very complex problem. Modeling biology is not easy. It is extremely complex. You put that into a mathematical model, and then you try how far your mathematical model goes when you're trying to reproduce biology. And biology is not straightforward. This is just a little. Oh, sorry. I still don't get the hang of this. Okay. You know, a bird or biology to a physicist, I mean, if you see a bird, that's just a bird. When you see biology, when you tell a biology person about the bird, well, you might look about, if you're talking about the peak of the bird or the wings, and they are different birds. And so we have to try to approach the problem not only from the physics point of view, because physics and equations, they're very nice. You can get all these equations and put them to solve, but you also have to attack the problem through the biology point of view. So why are we trying to do this? We want to try to understand the cardiovascular system, because even though we've been studying cardiovascular system for a long, long time, we still do not understand why some people suddenly get a cardiac arrest and then they die. We don't understand how are all of those mechanisms when you have a genetic problem or a genetic mutation, how will that actually affect you or not? We have no idea. We want to try to guide therapy because, well, we want to employ better all of the resources in medicine. We want to be able to help people heal. We want to, for example, make drugs safer for a particular individual. There is one drug that might help you, but that same drug in another person might actually kill them. And how we specify which person can use that drug or not, it's a very complex problem. And well, we want to try to understand that problem in a better way. Of course, we want to reduce the need for animal experimentation. We've relied on animals all along. It's not easy to do experiments in humans. It's not ethical, and it's undoable. And even though we use different animals, we've used rabbits, rats, dogs, pigs. You can use all kinds of animals to learn about biology, but they will never be humans. There are loads of models for mice. For example, mice are very easy to deal with because you can create mutations on them, and they grow fast because they have short lifespans, and you can create different diseases on them. But those mice will never be humans, will never really be the human characteristic of a particular illness. So we want to try to learn more on human anatomy, human physiology. Also for patient-specific modeling, this is a big trend worldwide. Now everything requires to be patient-specific because, yeah, we want to tackle the problem that one thing might help one person and not another one. But it's not a very easy problem. In fact, it's extremely complex. And well, but we want to try to learn how one particular drug or one particular thing might work well for one person. And for example, also for medical device testing and design, because when there's no much aid with pharmacology, you can get a device implanted, and that can help your heart keep on beating for a good while. It can help your heart from arresting. That's what happens to many football players, for example, that they get defibrillators implanted, and even though they might have a defect and suddenly have an arrest, they might make their heart pump again and stand up and keep on playing. So we want to try to address all of those issues. So as you understand, those are very complex problems. And so you got to start little by little. Anyway, who here works on heart? Anything heart? Okay, good. In cardiovascular vessels, arteries? No? Some other organ? No? Which organ?
Bones.
Oh, bones. Those are cool too. Really, a lot more complex than they seem. But anyway, okay, I'll give you a very, very brief description of the function of the heart. So overall, the heart is a pump. Even though people say you love with your heart, that is actually not true, because your heart, the only thing that it does, it just pumps blood. And it keeps the rest of your body alive. And well, it consists mostly of atria and ventricles. It's 4 chambers. You have the pulmonary arteries, the pulmonary veins that come all the way to the atria, pumps to the right ventricle. Right ventricle goes towards pulmonary arteries. Blood gets oxygenated, goes back to the left ventricle, and then it gets distributed to the rest of the heart. So one important thing is that it is a pump. So its job is to pump blood. So, however, how it does it, it's quite particular because it has an electrical or an electrical stimulus that makes the beat go in a particular rhythm, and that keeps it pumping all the way. So that's the part that I'll be focusing in mostly. Alfonso will talk more about the blood flow and the fluid-solid interaction problem. But know that this is mostly a pump. How the electrical activation occurs will affect how that blood pumps out of your heart. So how we divide, or how we model the full heart function is by dividing it into small little parts. So we start, for example, with the electrophysiology. So electrophysiology basically starts from ion channels. They are ions that go in and out of the cell membrane, and that ion movement in between the membrane can create a voltage gradient. So that is what we determine as electrophysiology. So that happens in every cell, in every myocyte. The heart is composed by, the cardiac cells are the myocytes, And funny enough, like there are more non-myocyte cells in the heart than myocytes. There are more fibroblasts. There are other cells that are all around there in the tissue as well, but they're smaller. But there are many of those. So the heart is not only composed by myocytes, but also fibroblasts, other cells that interact in between. Then to form the myocardium, the myocardium, as Eva mentioned, it has fibers. Basically, all of these cells arrange in a particular way so that when they contract, the pumping is effective. So there's been a lot of study about how the arrangement of these myocytes. So, and then you have the ventricles, the full heart. You have the propagation of the activation. This shows some of the Purkinje system. You have very fast conduction systems, so that most of the myocardium activates synchronously, so that it creates the best output. And well, then one thing you see out from the heart, like you can measure an electrocardiogram from whenever you go to the doctor and they put you some transducers, or how do you call them? Well, the ones that just measure. And you can look at the electrocardiogram. But this is all part of electrophysiology. You look at the gradients of the currents passing through the heart or acting in the heart in time. So from the mechanics point of view, there is a very tight coupling because the cells create the contraction. The cells depend in the ion channel motion, and the ion channels that are mostly related to the contraction is calcium. Calcium is a very important ion that triggers contraction in the sarcomeres. So inside the cell, there are these structures that are called the sarcomeres that they actually make the cell contract. And so they are within the cell, so they are also arranged within this fibrous structure that makes them orthotropic or anisotropic, this structure, and well, that will relate to the actual pumping effect of the heart. So up to now, this is more or less how we divide or we subdivide the study of the modeling of the heart. So I will talk a bit more about this part, the electrophysiology. A little bit about the excitation-contraction coupling, that is exactly this effect, or the function between the calcium and the sarcomeres and how they contract. And anyway, I'll continue a bit with that. So the action potential, that refers to the transmembrane voltage as you measure it within a cell. So if you measure inside and outside the cell, you will see this kind of curve. And that curve is, you can see, well, it behaves that way because of the transit of different ion channels at different times. And that is basically the biggest complexity about modeling electrophysiology. There are different ion channels that will let sodium currents, potassium currents, calcium currents in and out at different times. And all of the the orchestration of all of these ion channels create this action potential. So in order to model electrophysiology in a more detailed way, many of the ion channel or the ion or cell models include all of these or many of these different ion channels into their formulations. So each of them will have a few ODEs describing their behavior in time. Now, what is very important here is that genetic mutations that produce cardiac arrests or that produce other diseases occur within different channels in some of these molecular identities. So these ion channels are actually proteins, and the protein motion creates protein motion, and the interaction with the ion channels makes those ions go in or out the cell membrane. Well, how those proteins behave, how those proteins actually move and interact, that can define that there might be a genetic mutation, and if a particular protein is not moving that ion channel well enough, then that might create a problem, and you will actually see that problem in changes of this curve of the action potential. So many times in electrophysiology, what you look at, it's all of the changes that occur within the action potential. If it's longer, if it's shorter, if there is a bigger peak here or not, all of those things are related to the activity of different proteins in the membrane of the cell. Now, to make the thing a bit more complex is that not all of the cells of the heart have that exact same action potential. There are different ion channels that are active in different locations, and so that determines that different cells in your heart have different action potentials, and therefore they have different behavior, not only mechanically but also electrically. And if you want to try to model as closely as possible, many times you have to take into account all of these heterogeneities. One of the heterogeneities that is mostly modeled by most of the people that do cardiac modeling is transmural heterogeneity. That means within the wall, if you look at it transmurally, there are some behaviors or some phenotype of the cells that are endocardial, are defined as endocardial, mid-myocardial, and epicardial. So transmurally, the cells have a different action potential. They also have a different action potential, apex to base. And all of what that means is that there are different protein expressions of the cells in different locations. You also have here the Purkinje system, the Purkinje fibers. These are quite important in the way that they are fast pathways of conduction. So if you want your pump, the heart to pump in a synchronous manner, like in the best way possible, the conduction has to go quite fast all throughout from the AV node to the rest of the ventricles and activate as synchronously as possible so that everything contracts at the same time. So it's an interesting problem, all of the electrophysiology. So this is just a simple action potential little movie of the activation within a geometry of rabbit heart. Well, that is actually including the mechanics and some of the, and the fiber orientation description. And, well, it's just a little video. Anyway, so, I hasn't finished. That's it. So, the fibers, Eva mentioned them a lot, and for electrophysiology they are very important also, because the activation goes faster in the direction of the fibers than in the transverse direction. So we model it that way. Generally, what people have more or less measured is 1 to 3. So if you have a diffusion or a conductivity on this side, on the direction of the fiber, you have that conductivity divided by 3, more or less, in the transverse direction. Well, and some of the uncertainties of the modeling then start appearing, no? But that is more or less the convention. There's another thing about myocyte fiber orientations. These fiber orientations, they change also throughout the heart. There was one paper from, I think, 1960, where a guy measured more or less how much these fibers were changing transmurally in the left ventricle of a dog, I think. And he created a mathematical model of that gradient of that change. Sorry, no, not that one. Of how these fibers actually rotate more or less from endocardium to epicardium. So he wrote a paper. It's very famous because everyone cites him. He's Streeter. I don't even know his name, but his surname is Streeter. And so he described that the fiber angle changed from endocardial to epicardium from around 60 to minus 60 degrees. So he did that, measured it in the left ventricle of a dog. And since then, many people have adopted this model, and you actually can artificially recreate that change of the angle of the fibers transmurally in the heart. There were afterwards, in 1990, I think, no, 2005, there was other authors that measured also the angle orientation. This is by Anderson. And they measured this kind of a curve that can go from minus 90 to plus 90. Here you have to take into account that here you have the epicardial and endocardial and endocardial and epicardial. So they're about the same, but just this is more of a linear interpolation, more or less. And here they propose more of a cubic interpolation, more or less, in between the endocardial and epicardial fibers. Well, one limitation of this is that it is a good description for LV, more or less, LV transmural wall. However, it doesn't really apply for right ventricle. It doesn't really apply that well also for the base and the apex of the heart. So it's a big assumption. But many people use these models to create this kind of fiber structure. This, I think, it's fibers reproduced with Streeter model. So what happens now, most of the time, is that we want to try to actually measure these fiber structure in hearts, either the human heart or whatever animal heart you're modeling. And for that, people use magnetic resonance. Is diffusion tensor imaging for magnetic resonance. And you're able, what it does actually, it looks at, it relates the gradient, the biggest gradient of water molecules within the tissue. And that is correlated to the fiber direction. That also has its error things, you know. You're correlating water diffusion with how the tissue the fibers in the tissue are oriented. And I went back to the paper, and I think sometimes you can get up to plus minus 30% error. So, but that's as best as we can do. You can do that, or you can do that through histology, which means going there, cutting the heart, and measuring all of these angles, or doing micro CT reconstructions and trying to measure all those angles. And it's not easy. It's a very complex problem. And your conduction and your contraction depends on those fibers. So, you know, we're trying to approach patient-specific simulations. We're trying to approach, you know, very complex problems, but you have to be aware of all of the difficulties and all of the parameters that come into place when you're creating all of these models. So this is a sarcomere. This is the contracting unit of the heart cell. And I won't go much into detail. These are very important because they actually regulate how that contraction occurs. I'll show you a little bit of a thing, how it occurs. But the interesting thing as well is that sometimes these little heads, myosin heads, might not be at the exact best angle for it to move. How they do it, let me show you in a little bit, in the next one. How the cell actually creates force is by the attachment of this thick filament that has this little head to the thin filament. And when they go and attach, this is called the cross bridge. But how they do it in somehow like if they were rowing. So if anyone rows here, you may know that many times the way you put the, I don't know how they're called, the little pallet down and touches the water, it makes it more effective or less effective. Well, what happens many times with genetic mutations is that some of this motion between these little heads and the attachment to the thin filament might not be great, might be slightly slower in detaching, faster in detaching, and that actually can produce very complex heart diseases. Hypertrophic heart disease, I think it has a big component of mutations at the sarcomere level. And those are diseases that easily kill a lot of people. And so if you want to try to understand all of those processes, you want to try to model all of these details in a very detailed way. So overall, all of those things put more or less together will generate the cardiac cycle, more or less, that this is in the hemodynamic point of view. So that will create all of this pressure that is essential for everyone to be alive. And of course, all of the pumping of the blood from each of the chambers. So it's a really interesting, and not only that, I mean, then you have the arterial system where then the blood flow goes and gets distributed everywhere and it nourishes the rest of your body. So it's a very interesting, very complex problem. And there are many, many factors that are not trivial about them. But as far as you more or less know what you're dealing with, when are all of those errors, measurement errors, or all of those assumptions on your models, then you can better understand and interpret what your results mean. So as an electromechanic model, as I told you before, you have to put all of that, or as much as you want, or as much as you need from the knowledge of the basic physiology into mathematical models and put them into your code. So for example, if you want to have very detailed ion channels, there are different kinds of OD or ODEs, or different kinds of approximations for simulating an ion channel. If you want to be very detailed and to include genetic disorders, you may want to include some Markov kinetics in that, so be able to interpret how on each step how fast or slow that ion is going, how easy is that, how easily is that going on. And of course, you have to include different ion channels if you want to be very specific about, if you want to answer questions about electrophysiology. You can also include more simplified models of electrophysiology, basically an activation that resembles the action potential. If you want to look at just some dynamics of how that potential is traveling. So there's always this mix and match. You can put as many detail as you want, or as you can, because these become, after a while, they become very, very costly computationally. And of course, the material properties that we talked before, you have to include these into a finite element mesh. We use finite elements. And it depends how detailed or non-detailed or simplified your mesh is. And then you create some, you can reproduce the electrical propagation. In the mechanics, it's exactly the same. You can include very detailed calcium motion within your cell. You can include very detailed myofilament kinetics to actually include all of those rowing effects, and in the best or in a more detailed way to understand perhaps genetic mutations. The biomechanics, this is from a different model from the one that Eva showed you before, but you just can use different kinds of models for the mechanics, as you saw before, and of course boundary conditions. Because the heart will be pumping against a pressure, and it has to exceed that pressure so that it actually creates an ejection. So anyway, that's more into the modeling side. I will go very fast with this. Perhaps you don't really need to know a lot of the details. As I told you before, electrophysiology relies on this ion motion. So ions across the membrane can be transported by active transport or diffusion. And there are there are equations that can relate that ion transport in and out. So here, let me show you just a little movie so that you more or less get the point of this. So this is the point where molecular mechanics or molecular dynamics could potentially join. Oh, I'm sorry. Yeah, that is my dog. That is not the movie that I was putting there. Give me a second. Why isn't it there? A la otra, vale. Wait. Let me stop it. I'm sorry. Well, I did it on purpose. I always have to show my dog, to be honest. Okay. Sorry. Now, this is more or less where molecular dynamics can join or can have a little link with the kind of continuum assumption that we do when we're modeling all of these electrophysiology. This is an ion channel, and that is a protein that is letting in some ions. So when you're modeling all of these different ion channels of the cell, what we intend to do is actually interpret that mechanism of ion transport. So, of course, we cannot go all the way from trying to solve a molecular dynamics problem and then go up to the full heart problem. That's it. That would be, no, what are you doing? Sorry, this didn't really work that well. That's it. Stop. Okay. I can put this back. There. Anyway. So instead of doing that, doing the assumption of the molecular dynamics, you can more or less describe the ion channel in a more simple way. And this description of the ion channels actually gave the Nobel Prize to John Eccles and Hodgkin and Huxley in 1963 because they were basically modeling, before they knew much about molecular dynamics and before they knew much about ion channels, the actual behavior of ion channels, they described the motion of ions with some equations, with some time-varying conductance and ion and a current source, and to model what was here was the nerve cell. So the same basics for the nerve cell apply for the cardiomyocytes. The main difference between nerve action potential and cardiomyocytes is that nerve action potentials transmit information, and they do it in spikes. The cardiomyocyte, it has a spike, it has to transmit some information, but it also has to create contraction. And that lengthening of the action potential and then all of the traffic of calcium in between is what creates the contraction, and that's what makes them different from neuron cells, for example. It's mostly that part of the contraction and the lengthening. So this is the O'Hara-Rudy model. This is the first model made of human ion channels. So as I told you before, all of the models before this one, most of the ion channels were measured in animals. There was one model that it was interesting that one of the ion channel behaviors was measured in eggs of a frog to capture the dynamics, and then you more or less use those same dynamics, but change the current to fit what the current of a human could be. But this was the first model that actually included motion of ion channels in human cells. So this is the model that we're using at the moment. It has loads of different currents and calcium dynamics. And this is kind of like the state-of-the-art model that we're using. So when you move, well, I won't go that much into detail in this one now. I guess we won't have that much time, but you can derivate all of the diffusion electrophysiology or the diffusion of electricity in the heart by parting from cable theory. So basically, you assume and create all of your equations, assuming that you're having all of this current flowing through a cable. And from there, outside of this cable, you would have your cell membrane, more or less. We would have a conductivity, capacitance. And well, when you formulate all of this, and I won't go that much into detail, but if you're interested, you can always find it somewhere, I guess. Let me go down. So you're assuming that a current is passing through a cable that has a particular surface to volume ratio that comes here into the area and the surface of this cable, that this becomes an important parameter afterwards in the diffusion case or in the diffusion problem. And you have the capacitance. You assume that the cell capacitance is the one that is around that little cable. And you have an ion, you have a source of current within that cable. And the source of the current within that cable are the ion channels. And this particular part, the ion channels, are the ones that refer to all of the ion entities that I showed you in the cell model before. Here you have the diffusion. That diffusion basically says how easy it is for the electricity to pass through that medium. And, well, you can calculate your voltage gradient through that. So the equation that we're solving within the finite element problem, you have an ion channel that depends on your cell model, and you can have it as complex or as simple as you want it. You have your your membrane capacitance. This membrane capacitance, for example, also changes when there is you have an ill heart. For example, if you have the cells are swollen, the capacitance of those cells will be different, and that will affect the conduction within the heart. Then you have the diffusion tensor that represents also the anisotropic resistivity to the flow of the electricity, say, or the electric propagation within the tissue. And well, for the ion channel, ionic models, there are different models out there. If you want to model a rat, there are rat models, there are dog models, there are pig models. Well, pig I think I just saw once now. Or there are now human models. We're mostly using human models. And they can go up to solving 87 ODEs at each element. So that can become very, very big, very, very quickly. So when you are defining also your problem within a ventricle, many times it's very important to take into account the Purkinje fibers that I told you act very quickly to transmit all of the activation within the heart. And many times the assumption is you can activate all of the hearts at the same time. It's not the best assumption, but when you don't know exactly where are your Purkinje terminals, because this is a very complex network that goes within subendocardial regions, and it actually just connects to the tissue at particular junctions, at particular ending points. So when you see this network, it doesn't really propagate through the network to the tissue. It just goes at different points, and where are those points located is a very difficult problem to know. So as I told you before, this is more of the electromechanical description. So you have the action potential. The action potential makes ion flows from inside and outside to the cell of the cell. And then you have the calcium current, that the calcium current will trigger contraction. And then, well, it will trigger this contraction. There is a bit of a delay, for example, in between the peak of the calcium current and the contraction. And this is due to all of the actual mechanics of the sarcomere. And this is easily modeled, the model that we have been using up to now, we're hopefully starting to use a new model now, is some just forced calcium relationship. So we're not taking into account of how these sarcomeres are moving in and out, or binding and unbinding. You basically create a force based on a calcium concentration. This is one of the simpler models you can use for excitation-contraction coupling. This model, this comes from a publication from Hunter and McCulloch in 1998. And it gives you these loosely coupled excitation-contraction coupling, where this active stress is actually related to the concentration of calcium within the cell. And these are some of the parameters for these activation-contraction coupling. But it's basically, a force-calcium curve relationship. This relationship can become faster or here by changing the lambda. So you might be able to create faster force, slower force, depending on these parameters. But these parameters are still very phenomenologic. They do not really represent the sarcomere binding, well, the sarcomere function directly. So, but this is what we have done for now for excitation-contraction coupling. We're hopefully trying to use a newer model, but anyway, this will hopefully come soon. Passive stress, Eva has told you everything about it, so I'll just skip that. And so beforehand, there was a very interesting question about how do you validate these models and how do you make sure that your constitutive solid mechanics model is good? How do you make sure that your electrophysiology is good? How do you make sure that your contraction is good? It is very difficult. And it is an active problem that it's not fully solved yet, I would say. There are many ways to try to validate different parts of it. But when you have a multiphysics coupled problem, the addition of these parts of the validation of these parts might not exactly be a full validation of the problem, because it involves different boundary conditions. It involves different hearts with different behavior, active and passive behavior in terms of the contraction. It involves hearts with different phenotypes. It might have mutations. It might have an infarction. You know, you have all of these issues that are very, very complex. You have to make the most with whatever you have. Whenever you can have measurements of, for example, echocardiography, you can have information about valve function. We haven't talked about valves, but valves is another big issue. And it's very difficult to solve computationally, but it's also very difficult to try to validate all of that. Functional information is very important. Any hemodynamic parameters, ejection fractions, strains, torsions. CT and MRI imaging are the two modalities that people use the most. You have to be able to look and to segment a heart, the heart, in the best way possible. And if you have the heart in systole and diastole, that many times you have with cine MRI, you have the actual function, you can more or less relate how the heart that you're simulating is contracting with what you're looking at in the cine MRI. Pressure measurements are not very common to find, because the only way to measure pressure is to be invasive, mostly. And looking at left ventricular pressure, right ventricular pressure, or aortic pressure is many times completely out of the question, unless you're doing an invasive catheterization of the patient. Then you can actually have these measurements. Left ventricular pressure is extremely difficult to obtain, but it is many times desirable, because that left ventricular pressure is part of the boundary conditions to your electromechanic modeling. If you do not have a fully fluid electromechanic problem solving. So it is highly complex. Most of these validations have been done more or less in animals, but there's still a lot of work to do with that, because computationally it's very expensive. It depends exactly what you want to validate. If it's mostly the mechanics, many times you don't have enough of mechanics and electrophysiological measurements at the same time. So it's complex and interesting in any case. You have the electrocardiogram that you can also try to solve a full torso problem, where your heart, the model of the heart is embedded in a torso, and then you also solve the diffusion of that gradients of the heart into the torso up to where you can measure the ECG. It's also extremely computationally demanding, but it's also a way to kind of inverse problem in which by looking at the ECG, at different ECG leads, you can more or less understand where the heart is activating faster, or first, where are the initial activations of the heart and the latest ones. So there's a lot of work that can be done with that. There's electroanatomic mapping that doctors do routinely when there are patients that go when they have either atrial fibrillation. Doctors go in and try to measure how is the voltage and where are rotors of activation happening, and then they go in and burn the site so that those rotors don't happen, because those create atrial fibrillation, for example, and that is the same with the left ventricle. Doctors go and put in transducers, and you measure at different locations all of the voltages, and you can more or less use them to parameterize and somewhat also validate your model depending on what kind of thing you're doing. Perfusion information, well, there's contrast MRI. It's done a lot to look at regions of the heart where there have been infarcts. So you have a scar. It's a region where conduction will potentially be very low. There will be not a lot of contraction. And if you want to look at, to model that, to see what is the overall effect, to see if there are any conduction channels within that scar tissue that might actually create you ventricular tachycardia or other reentries, doctors have to know more or less where are the locations, what are they doing, what is the status of the tissue there, if it's alive, if it's more or less alive, if it's ischemic. And all of that changes your electrophysiology, and it also changes your mechanics. So it is not, it's a very complex problem. And anyone who does more work on this and that, and trying to validate and set up all of the benchmark problems and all of that, it's awesome. It's particularly welcome. And now, I'll just go very quickly on some of the applications. This is a very cute, well, it's a nice application because it's on drug safety testing. And this is the application that I showed before. There are some drugs that might actually create more of a problem to a particular person than solving their issue. There's a lot of thing about, there's some drug, when you develop, or when pharma companies develop drugs, first they take a long time. It takes a lot of money, and most of their studies, they do them in mice, in cells. Now there's a lot of work in pluripotent stem cells. So some of these cells, what they do is like they take samples of cells from tissue of a particular patient, they grow them in dishes, and they make protocols so that they transform somewhat into myocytes. And then what people do now is you apply drugs to these myocytes and see how they are behaving. And somewhat you want to translate that in the actual behavior of a cardiomyocyte in a patient. So there are different ways kind of people are trying to solve the problem of drug discovery, but in any case, it is still very costly. It relies a lot in animals, and it's still not fully solved. So what we attempt to do, well, and as I told you before, many times all of these drug testings are made in rabbits, rats, mice, whatever. And then they want to look at how much can that a particular drug can create an arrhythmia in a heart. So the state of the art on this is that they take the heart of a little dog, take it out, cut a little chunk of the tissue, put electrodes, infuse, keep it alive, infuse drugs, and see more or less how they are behaving. And this is a little footage of one of those tissues. And this is mostly how people do drug testing, cardiac toxicity. So basically, to this little chunk of tissue, they keep it perfused and they put drugs. And then you can see that then you pace that tissue. And then you see at some points that particular contraction is not very regular anymore. So then you see that it's creating a bit of an arrhythmia. So what people do is like they put electrodes and measure the pseudo-action potentials on different parts of the tissue, and try to see how much drug induces arrhythmia, and or if a particular drug has some potential drawbacks. So what we're trying to do is to recreate that computationally, and they have different protocols of stimulation to look at arrhythmia. They're called these restitution curves. So basically it looks at the dynamics of the action potential when you start pacing it at different frequencies. And you put you have it pacing at a particular frequency, and then you cut the time in which you pace it the last beat. So you see the dynamic response. And based on that, there are these restitution curves, and you can measure how arrhythmogenic a tissue is. So I've been working to try to create this in wedge preparations in humans. This was some initial work that I did before with using a model of published before by Tentuscher, the ionic cell model. And well, we're trying to make better simulations for this. But the idea is that, I'll go farther. The idea basically at the moment is to recreate this. This is actually a human ventricular wedge preparation. I forgot to put the reference, but this is from Professor Efimov's lab in Washington. But what they do is they have a human wedge preparation. Many times when there are humans that give their organs as their donors, and if there are no recipients of those hearts, and they still and they still want to donate that heart for research, there's different places where they can receive the hearts and do these kinds of experiments and basically try to understand better either an illness, or try to understand the response of the human heart to particular stimuli or particular drugs or things. So what they do is they have this little piece of tissue, the same thing that you would do with the dog, and put electrodes, stimulate. Here what they do is they also inject dyes, and these dyes actually this technique is called optical mapping. So basically you have these particular cameras, very high resolution cameras, that will capture how the dyes will glow depending on the different dynamics of different things. You can dye for calcium, for example, so you can see the wave of calcium propagating, or you can see the calcium transient at different locations in this heart, or you can see the voltage gradients. The idea now is to obtain some of these measurements and parameterize some of the O'Hara-Rudy model to be able to recreate this kind of activation. This is the activation of a normal heart. This is the activation and the action potential duration distribution. So as I mentioned before many times, how you measure all of these ion channel dynamics is by looking at the length of the action potential. So it's an important parameter that we use for the electrophysiology. So this is some work, it's still underway. This is just, this is my human wedge. This comes from a high resolution MRI segmentation. And what I want to try to do is to recreate the human wedge preparation for electrophysiology simulations to somewhat try to validate the model. But this is still kind of ongoing thing. So one thing I want to point out is, like, many times when you see the or the cardiac models, you see these really nice smooth surfaces, and that's really not the case. That's really not true. The heart is far from smooth, and this is something we've been working on. The heart has a lot of muscles inside. There's loads of trabeculae, there's a lot of papillaries. You actually have little, these are called false tendons. They're basically cables connecting electrically one side of the heart to the other. And all of these things, I mean, we normally, or in simulation, you normally take away because they're very difficult to model. One, to segment from medical images. Many times when you have a medical image, you have it from a human that is alive, and the heart is moving all the time. So trying to obtain a good resolution from a moving heart without leaving the guy there burning for 24 hours, it's impossible. These we're getting from ex vivo donor hearts at high resolution. And there are loads of different problems that haven't been solved really of how all of these structures are important and how they interact to either create more arrhythmias or maybe not, or actually to create a better conduction. Sometimes when these little false tendons break when doctors are introducing things, they might actually completely change the activation within the heart. And that might actually make a huge change, huge difference for the patient and the whole pumping. So there are all of these issues that we're trying to tackle. I don't know if I'm going to be able to go much through it, because we have started with hemodynamics, analyzing hemodynamics. I'll skip a little bit this. We've analyzed some of the wall shear stresses of that, but that's a completely different hemodynamic problem, so I'll kind of skip about that. This is also wall shear stress in the right ventricle and left ventricle. Right ventricle has whole loads of these structures, and many times when people do electrophysiology or hemodynamic studies, they completely take out all of those tissues. And we believe it makes a huge difference. So let me see. In terms of vorticity, and when you have a simulation of hemodynamics without the structures, these are the vortices. And you can completely see the difference when you have all of these structures coming in between the way of the flow. These we still do not have any electromechanic simulations, but we're trying to move towards the electromechanic simulations. That's how different the wall shear stress is when you take into account that all of the trabeculae and papillaries and muscles in there, and when you actually take that away, and then it's completely different. So many times when you're doing all of these simulations, it depends, one, on the question, and two, well, how much do you really want to understand from the problem? Or our point of view is like, well, we need to push a bit more the boundaries to understand what is important and what is not, when you try to extrapolate those measurements to a real human, or to try to do more of a predictive thing. So I'll skip a bit this, because this is mostly flow. And this is mostly, we're starting to do this on electrophysiology. So again, when you have this problem, you have this really complex structure, you need to define fibers in it. We have been unable until now to find high resolution DT MRI, because mostly you cannot, from the resolution of the DT MRI you can get now generally, you cannot really extract information from trabeculae or papillaries, even ex vivo. But we're trying to get more of that. These are made from models. Actually, on the trabeculae and papillary, we're actually generating those fibers by following the main pathway of the trabeculae. And then you applied some modified Streeter model for the rest of the myocardium. In this case, we're collaborating with some people from Pompeu Fabra, which they created a model for particularly the right ventricle, because the right ventricle is one of the cases where you cannot possibly rely on a Streeter typical model, because the fibers are completely different. So anyway, this is something we're working on. This is from my student Federica Sacco. And this is still a toy problem, a toy model. There's a lot to be done there, but we're trying to move into that and comparing with the smoothed geometries that people generally solve. This is how it is done mostly, because one, you do not need that many more elements to describe this smooth surface. And second, you do not have that much information to extract all of the endocardial, the little trabeculae and papillaries. But, well, we're trying to push it further. And overall, what we attempt to do is actually to run more of the electromechanic problem with all of these structures. So you can imagine, like, to run that is in the full heart with all of the trabeculae, papillaries. That's going to be a bit of a challenge. Computationally, you do require a lot of resources. At the moment, we generally solve, well, we do solve only electrophysiology at the same resolution of the mechanics. So we have our model. You can create fully coupled models. You do not have to do any interpolation in between the mechanics or the electrophysiology. It depends on the model of electrophysiology also that you use. You might require solving, I think the O'Hara model is about 41 ODEs at each node, and then you have many millions of them. And then, of course, you have the mechanics. But, you know, that's why there are supercomputers, and we need to be able to use them. So that's kind of the thing that we're aiming for at our department. So there's just one more thing about this problem. I think in terms of electrophysiology, having endocardial structures, all of these things make a difference. They impact a lot of all of the electrophysiology, but I think they will impact more the mechanics. There have been studies in which they look more or less at the distribution of all of these muscles inside, and they've seen more or less of sometimes a helicoidal distribution. So if you actually take into account that those muscles do contract and do create a lot of force, they might actually help a lot, one, in torsion, and second, also in lowering of the base when the heart contracts. And those are some of the things that are, I mean, I've seen in literature, it is very difficult to obtain the full torsion and the full contraction of the heart, of the simplified smooth geometry. And actually on those geometries, many people have given up using the DT-MRI measures and started using more of a plus minus 90 degree transmural distribution of the fibers to compensate for that, to compensate for the actual longitudinal contraction. And well, we have the theory that most of that force is due to all of these structures that you generally take out because it is just too much to try to solve. But I don't know, we're trying to figure that out. So perhaps in 1 or 2 years of the PATSI, you might come and we might have an answer, but we still don't know. So yeah, that's more or less it. I don't want to extend, I have loads of other stuff, but I don't want to extend too much. I was given about an hour, and I think it's about that. If there is any doubt or question, no? There's just one last ad that I want to do. We didn't have a hands-on on this workshop, but there is going to be a VPH summer school in Pompeu Fabra, and we're actually going to do more of a hands-on of electrophysiology, if anyone is interested. And that will be actually really interesting because you will have to, the students will have to extract fiber orientation from synchrotron data, synchrotron radiation data, micro-CT, so to run electrophysiology simulations. But anyway, that's the last thing. And the work group, including the dog. Yeah, thanks. That's it.